So thank you very much for having me today. And as it's just been introduced, I'm going to be discussing developing a platform process for LNP manufacture. So a little introduction to my company first. So we are a UK-based company called CPI, and we actually sit within the innovation space in terms of drug discovery. So we don't sit in the drug discovery section of the research and invention, and we're not quite in the commercial market either, but we sit within that middle space where we're trying to help UK industry customers try and get their products from research and invention to commercial market using both public and private collaboration projects. How we do that, we do a lot of proof of concept and scale-up studies, so to prove the feasibility of new ideas before approaching investor stakeholders funding programs, reduce the risk of scale-up as well. So we have a lot of facilities here at our different sites. So it may be that before investing, you'd want to be testing some of our facilities first, so that helps you to prove and refine on novel technologies before investing further, and decreasing time to market, so trying to make everything much more quicker and accelerated throughout the process as much as possible. And the areas that we focus on are these ones on the screen here. So I sit within the pharmaceuticals, but we've also got medtech, agritech, FMCG, logistics and packaging, electronics, energy, specialty chemicals, materials, automotive and aerospace, as well as food, drink, and nutraceuticals as well. And where we're based, as I said, is in the UK. So we are in the northeast of England predominantly. So I work at the Sedgefield site, and we've got another couple of more sites around that are only 20 minutes away, half an hour away. We've also got a new site up at Glasgow, which works on powder pharmaceutical preparation. And we've got a couple of offices around the UK as well, including Newcastle, Manchester, Liverpool, and Cambridge as we're expanding. So my job is to work within the nanopharmaceutical and nanomedicines area at CPI. So we work from a formulation point of view. So we work on the process, so looking at the in-process controls, how that affects the critical quality attributes. The biological performance is outsourced, so that's not currently sits within CPI. But as we all know, that these nanomedicines can treat previously untreatable drugs. And with that comes a much more refined and sometimes more difficult process in process controls, etc. So we work along that stream, helping companies to be able to refine their process and scale up to get into commercialization. What our process looks like. So this is the complex medicines process that we generally follow. We've got the manufacturing process, which covers self-assembly across multiple different mixing technologies. So, for example, microfluidics, homogenization, etc. We also focus on the purification, so by batch and continuous TFF, isolation by lyophilization, and then we outsource the in vitro and in vivo testing. We have a lot of characterization capability that sits across those different process steps, so including particle size, surface properties, structure, loading, chemical composition, makeup, release rate, and product stability. And this covers a range of delivery vehicles and APIs as well. So there's someone on the screen there, so including liposomes, LNPs, polymeric nanoparticles, inorganic nanoparticles, and biological pharmaceuticals, including nucleic acids, and also small molecules as well. So a little bit of background as to how we got into this area. So being the formulation department, we've got a core expertise in colloidal formulations. So we got involved in this project here about 2016, 2017, along with a multitude of partners, including AstraZeneca, Pfizer, Malvern, Precision Nanosystems, Croda, universities, Manchester and South Clyde, and the NIHR. And the aim of this project was to create a UK hub, basically, of a pilot scale hub to be able to produce different nanomedicines, so for use in the treatment of cancer or for vaccines, etc. And what this looked like was this pilot line here. So it was using the Precision Nanosystems Blaze to combine two phases, one organic and one aqueous, and then a custom-built continuous TFF system to be able to clean that up. So this is where you'd be looking to do any sort of buffer exchange, solvent removals, concentrations, etc. So it's a one-pass system with a Malvern Panalytical Zetasize AAT, which is the outline particle system to give you a reproducible product every time. So this is a pilot line that is available mainly to support the UK industry, but also for for customers outside the UK as well, to be able to scale up the hard to process and hard to scale up the systems already. This left us into a very good position just before the pandemic. So there was a need to develop some UK capability for RNA LNP vaccine manufacture. So we supported Professor Robin Shattuck's team in terms of tech transferring and scaling up a process for saRNA synthesis, purification, and encapsulation. So we've got a biologics facility that works in close partnership with our formulation facility. So the biologics facility focuses on the RNA, and the formulation facility focuses on the LNP encapsulation. So this had the intention of going into GMP manufacturing, but it didn't get quite that far through the clinical pipeline. But we managed to set up a lot of capability and kickstart the GMP facility setup in Darlington that we've got right now. So where that puts us at the moment is we've got a lot of RNA and LNP R&D innovation capability. So this is counted as our consultancy services, both in fee-for-service or funded collaborative R&D projects, where we can either consult or we can carry out any sort of lab-based R&D work that companies need to be able to help progress with their processes and products. So this involves both the RNA, saRNA, LNP and alternative delivery systems as well for a wide range of cargoes. Then we've got two centres of excellence. So we've got one called the RNA Centre of Excellence Training Academy. So this is where our GMP manufacture capability sits. So we've got a GMP facility that is able to make RNA LNPs all in one building on one site. And that's to be able to help people get things through to the clinic quickly. So move things from R&D to the clinic for RNA synthesis right through to encapsulation. Part of that, there's also the RNA Training Academy, which is intended to be able to help upskill the workforce and drive industry skills in this area. Then we've also got the ICDC Centre of Excellence, which is the Intracellular Drug Delivery Centre of Excellence. This was previously called the LNP Centre of Excellence. But as it's developed, it's decided to open it up to a wide range of delivery vehicles. So the initial focus will be on LNPs, which is why you can see that they're on the slides here. But it is for any sort of nanoparticle delivery system, really. And the idea of this is to be able to work on a lot of the industry challenges with LNPs at the minute, so on high throughput screening and being able to create lipid libraries, etc. A little bit more building onto each of these centres. So the RNA Centre of Excellence, we've talked about the unique R&D capability across RNA LNP, but critically it's got an RNA LNP optimised manufacturing platform, which is what the bulk of my talk here is going to be about, how we develop that platform process. And I've talked about the GMP manufacturing facility, so that was funded by the UK Vaccine Task Force, and the training packages, which are just starting to be rolled out now. And the ICDC Centre of Excellence. So the idea is it's going to be a single front door for companies and academics to access support when developing these LNP-enabled nucleic acid therapies and vaccines. It aims to bring together leading UK academics and catapult centres. So you'll see some of the partners on the right-hand side there. So in addition to CPI, there's University of Strathclyde, Catapult Medicine Discovery, Imperial College London, and the University of Liverpool as well, that are all inputting into this centre. And the idea is to accelerate our knowledge and eliminate any key industry challenges in this as well. So coming on to the platform process. So this is the platform process that we've developed at CPI. So we've got the RNA and lipids encapsulation, followed by immediate inline dilution to be able to reduce the ethanol content. At scale, we run TFF, tangential flow filtration, to be able to clean up the product before we move into final formulation, final filtration, and then filling for storage at low temperatures. So the key thing that we've done here is looked into the critical process parameters needed to ensure that the critical quality attributes can be met across the batch. The internal capability projects that we run here have allowed for the development of this process and further understanding for improvements. And I'm just going to go into some of the experiments that we've carried out that's enabled us to fix this process in place. Some of the critical quality attributes that we look at, so including particle size and PDI by DLS, CT potential by ELS, and then we can outsource particle size morphology by cryo-TEM, In terms of the RNA itself, we look at RNA concentration, encapsulation efficiency, so either by UV-Vis or by the fluorescence assay Ribogreen. We looked at RNA integrity by gels, SESI, so capillary electrophoresis or HPLC, RNA identity, and then we look at the lipids, matrix, etc., sterility, activity by all the standard assays as well. So in terms of the microfluidic parameters, so we've looked at investigating the effective flow rate ratio on the particle size and PDI. So that's a key attribute to be able to make sure that you get right for the encapsulation stage, and also what the total flow rate is. So making sure that we have a scalable system is really important to us. So does it work at 5 mils a minute, 20 mils a minute, and does it work at even larger scales like 100 mils a minute for that scale up size? So we've looked into both of these parameters to make sure that they are optimal for producing the right particle size and the lowest PDI we can out of the encapsulation step. A key process parameter that sometimes gets forgot about, I think, at the early discovery stage is the increase in nanoparticle concentration. So in terms of scaling up, you want to be able to keep your volumes as low as possible. You want to reduce your waste. So for example, to going through from encapsulation to TFF, the smaller volume you've got coming out of encapsulation minimizes your TFF volumes as well. So this is a key thing for us in terms of scale up. So making sure that we can maximize the concentration of the nanoparticles to be able to minimize the volumes and make the whole processing easier. So we've done this by increasing the nanoparticle concentration and looking at the size effect, PDI effect, and also the encapsulation efficiency as well. And you can see that the particle size does increase as the nanoparticle concentration increases. So it may be that as you've got larger RNA, there may be a smaller limit to the nanoparticle concentration you can run, but the encapsulation efficiency remains broadly the same. Changing the dilution ratio post-encapsulation. So this is a key factor and it plays into those volumes again. So we want to be having a dilution factor post-encapsulation to make sure we dilute out the solvent or ethanol that we've got in there, because that can affect the particles, the stability of the nanoparticles. So wanting to maximize that dilution factor for the stability of the particles, but also minimizing it to be able to ensure that we keep the volumes low, especially before we go into the downstream steps as well. So it's about finding that balance between this step really. So making sure that you have a stable particle for as long as you need, but that they're not too large volumes that actually really affect your downstream processing stages as well. The addition of cryoprotectant. So I think it's quite well known now that the RNA vaccines need to be stored at -80 degrees C, so very cold temperatures. There's a lot of work going on to be able to create LNP systems that are stable at more like fridge or room temperature. But most of the systems need cryoprotectant in there to be able to store them and keep them stable. So where that cryoprotectant is added, it's usually added in downstream steps. So for example, dialysis, TFF or final formulation stages, but can you actually add them earlier in the system as well? So here we've looked at adding it in the encapsulation stage. You can see that it doesn't really have an effect on particle size, PDI and encapsulation efficiency. So we've tried to increase it to a maximum. And also looking to include more than one cryoprotectant, so 2 cryoprotectants, and see what the effect that has. And the scale up from milliliters to liters. So as you probably gathered, a key thing for our processes is they're able to scale up. And that's what we get asked most to do. So given a 2 mil system and then having to scale it up to the litre scale. So we use the Ignite and Blaze systems quite often within our labs because it enables a really quick and easy scalable process. You don't need to do much optimization, if any, to be able to scale it up. So here we look at the Ignite encapsulation stage and then taking that through to clean up using dialysis. So this is at roughly 1 mil scale, and then the litre scale. So going up to 1, 10 litres, etc., using the Blaze and cleaning up that using TFF. And you can see the particle size, PDI, and encapsulation efficiencies are comparable across both scales. So this is an overview of our platform process. Example batch results. So this does depend on what RNA LNP you're encapsulating or the batch size, but these are just some example results, and you can see we've got good particle size and PDI and encapsulation efficiency after the encapsulation stage. As we move down into the downstream processing steps, this is where you've got to really keep an eye on both your particle size, PDI, and encapsulation efficiency. It's not just about getting the good quality out of the encapsulation, it's about what it looks like at the end of the process as well. And you can see the particle size and PDI remain broadly consistent. The encapsulation efficiency does drop a little bit, which is expected, but still way above the specification, which is good. And you can see the particle size distributions that are looking consistently good across the whole process. We also measure the RNA and lipid concentrations to check we've met the critical quality attributes for them throughout the process, and checking the lipid molar ratios haven't changed as well. We also check the ethanol content at the end of the process. So have we actually removed all the ethanol from the encapsulation step through the TFF? What does our endotoxin look like as well, which isn't on here, but making sure we've got low endotoxin content. And really importantly, what is our RNA mass recovery? So it doesn't matter what you put in, it matters what you get out. So if you've lost more than half of your process during that, which is can happen sometimes, half of your RNA during the process, which can happen sometimes, it's not a very good process. So we've managed to keep the RNA mass recoveries above 80%. We've talked about the platform process in quite a lot of detail, but what does that really mean? So it doesn't mean that one size fits all. So this is just an example system where we've tried to look at the critical process parameters and the effect they have on the critical quality attributes. But ultimately, if you change a critical parameter, you're going to have to go back around the circle. So you're going to have to develop the process through research and development, establish the process, and then again, if you change something, it may have to go around that loop. So it depends on what you're encapsulating. Is it a different type of RNA, different sizes of RNA? Is it DNA instead? Are you changing your lipid system or the molar ratios of the lipid system? This may have an effect on the stability, on how it encapsulates, how it performs downstream, etc. Are you changing any excipients? So that's a critical one for stability. And also you're changing any of the equipment as well. So all these could affect what your process looks like. So even though we're saying it's a platform process, it's basically a starting point. It's expertise that we've generated, but it doesn't mean that it's going to fit everything that comes through the door. The example of this is looking at different lipid systems. So what we changed out here was between the D-lane MC3 DMA, so that's the Onpattro ionizable lipid, which is used quite extensively within research. If you just change out that ionizable lipid for different ones, which have got different pKa's, it may look sometimes quite similar during encapsulation. In this case, it doesn't. So it does actually change the particle size quite a lot. It may perform similarly during downstream processing, but also what does it look like after stability? And you can see here that the middle graph shows an increase after freeze-thaw up to around 600 nanometers. So all of these things can have a massive effect on what your process would look like. So this would need to be completely redeveloped for this lipid system. In terms of encapsulation of RNA at CPI, we've covered using poly-A quite often as a surrogate for RNA. So this allows us to look at the process and maybe develop some of our analytical techniques before we move to using the really expensive and easily degraded RNA. But we also looked at RNA of different sizes, so saRNA, mRNA, saRNA, etc. And again, all of these can have an effect on what your process looks like, especially those downstream processing conditions which often get overlooked. Other delivery systems that we've looked at, so we're not just on about LNPs, but we're looking at liposomes as well, small molecule drugs, inorganic nanoparticles, peptides for nucleic acids, and polymeric nanoparticles as well. So this platform process was designed for the LNPs, but because it's a complex medicine, it can be taken and applied to other different lipid systems, other different delivery systems as well. But again, you need a lot of R&D work to be able to go into that to make sure that you've got the right CPPs for the right CQAs. And in terms of what the future of RNA encapsulation looks like, so on the process side, a lot of the questions we're currently getting asked is how we can increase production, how we can increase the mass recovery, reduce any losses, and meet the needs of another future pandemic as well, and improve the analytical assays. So a lot of the process and analytics that were put in place, are they the best? They were developed during a pandemic to be able to get things out the door as quickly as possible, which was amazing. But are they the best for what we can develop? Is there somewhere we can go with that as well? In terms of increasing understanding, there wasn't a lot of understanding on the LNPs, especially to do with the structure, etc. So now a lot of people are having the time to do that research and use different analytical techniques and characterization methods to be able to understand those LNP systems, improving the efficacy and the endosomal release. So a lot of that work is going on at the minute, which is great to see. Unlocking new therapies, so looking at targeted therapies, previously untreatable diseases, etc., but also what the next LNP is. So LNP is kind of been turned to a buzzword at the minute, but what is the next one that's going to be coming along? Is it going to be a different type of LNP? Is it going to be a different delivery system altogether? And what advantages will it have compared to the current one? So that's it from me. So thank you for listening, and I'm happy to answer any questions that you've got as well.